welcome to the next module. In this module, we will be studying about antifreeze proteins part 1, introduction, structure and mechanisms. So the first we will be looking at introduction to antifreeze proteins and cycrophiles. Then we will move on to study about antifreeze protein structure and mechanisms. So the entire study about antifreeze proteins started by some curious observation which puzzled the scientist. When the prominent biological oceanographer Dr. Backus asked a simple yet a powerful question to his colleague Scholander at Woods Hole Oceanographic Institution, Cornell University, that Arctic fish swim about in ice water at minus 1.7 to minus 1.8 degrees centigrade. Why don't they freeze? Because the freezing point of the body fluids of these fishes were around minus 0.5 to minus 0.8 degrees centigrade only. So, do they have twice as high an osmite concentration as ordinary fishes, or what is the story? So this is how the study about antifreeze protein started in 1953. After receiving the question, there were some challenges faced by Scholander to find an answer. So he chose Nain in the northern Labrador as the place to go for his studies in the Blue Dolphin Labrador expedition in 1954. But unfortunately, the rain melted the ice along the Labrador coast and made an airplane landing impossible. So he has to choose or shift the study area to some other place. So then they tried to catch fish by chopping a dozen holes through 4-foot ice with the help of Inuits and a dog team. Of course, the freezing point determination can be done in a 10 to 20 miles from the base. So these are some of the challenges faced by Scholander, but he never gave up. Now let us move on to psychrophiles. So psychrophiles are basically microorganisms belonging to bacteria, archaea or eukarya, and they thrive in extremely cold environment, typically at a temperature below 15 degrees centigrade. These organisms can be found in various cold habitats such as polar ice, deep ocean waters, glaciers and permafrost. They play a crucial role in biodegradation and nutrient cycling, contributing to the ecosystem's health. Their unique adaptations enable them to withstand extreme conditions including high pressure, salinity and low nutrient availability. There are two major types of cold-adapted microorganisms, especially the psychrophile. One is obligate psychrophile, another one is facultative psychrophiles. So, when we look at the obligate psychrophiles, they prefer a temperature below 20 degrees centigrade. The optimum is around 15 degrees centigrade, but they are unable to grow above 20 to 25 degrees centigrade, whereas the facultative psychrophiles' optimal growth temperature is above 20 degrees centigrade. They can survive below 0 degree also at times. And when we look at the habitat, the obligate live in the polar regions, mountains, glaciers and deep sea. Some of the examples include Psychromonas species, certain Arctic bacteria. When we look at the facultative one, they live in an environment with a periodic and seasonal temperature fluctuations, and some of the examples include Pseudomonas species, some yeast and moles. If you look at the reproductive temperature, the obligate species, the minimum temperature for reproduction is not known, whereas for the facultative psychrophiles, the minimum reproductive temperature is around minus 12 degrees centigrade, but their metabolism stops at minus 20 degrees centigrade. So that means the optimal growth for them is actually above 0 degrees centigrade. The water availability is crucial for both the psychrophiles, the obligate and the facultative one. And when we look at the cold tolerance, the obligate one, it is very essential for their survival. They cannot grow at higher temperatures. So this is the major drawback with obligate. They cannot survive at high temperatures. Whereas the facultative one can tolerate cold, but they does not require it for their growth. They can adapt to moderate warmth. So that means this is the major difference between these two species. That is, obligate must live in cold environment and are specifically adapted to function at low temperature, whereas the facultative ones can survive in cold environments. And there are few changes, adaptations taken by the psychrophiles to survive in the cold, harsh conditions. One is at the molecular level changes, which include increasing the fluidity of the lipid membrane, synthesis of special protein, chaperonins, synthesis of cryoprotectants, reduction of growth factors, synthesis of cold active enzymes and metabolic changes. So these are all happened at the molecular level for their psychrophile to live in the cold environments. And when we look at the behavioral changes, they generally go for hibernation under mud, so they want to prefer at low temperatures, or the facultative one may migrate to warmer areas for their survival because they can tolerate cold but they cannot live for long in cold. And their annual lifespan also changes according to the seasons. When we look at the psychological strategies, they have two major things. One is called freezing avoidance, another one is freezing tolerance. We will study about these two things in the later part. Let us move on to the next important one, which is called the biological antifreeze. We had talked about the psychrophiles, but how do they survive? They actually have some specific chemicals because of which they could survive in the cold environment. So in the polar region, the temperature of the seawater is constantly sub-zero due to the existence of dissolved salts. These sub-zero temperatures are very damaging, if not fatal, to the polar organism. So this is the first question asked by the Bacchus. If the temperature is below -1 their freezing point of the body liquids, then how do the fish survive? So this is the biggest question the scientists asked. So later on, De Beers and Wolfgang have shown that as the cold variation mechanism, antifreeze glycopeptides or antifreeze proteins or peptides are produced by various organisms for protecting them from freezing injury. That means they can actually survive in the cold environment. So this is what people had found, and they gave the name biological antifreeze. These proteins are known as biological antifreeze, and there are two major classes. One is the antifreeze protein, another one is antifreeze glycoproteins. The AFPs are one of the main proteins that remain functional in cold temperature, naturally encompass bony fish, insects, plantae, fungi, nematoda, amphibia, bacteria and diatoms. So some of the important keywords we will be studying in this particular module and the next one are ice binding proteins, ice nucleating proteins, antifreeze proteins, thermal hysteresis, ice crystallization inhibition, supercooling point, colligative properties, nucleation temperature, hexagonal, bipyramidal or crystal shapes and basal planes of ice, Gibbs-Thomson effect.